Hello everyone, welcome to our webinar, These Trees Cast No Shade, Understanding HIV Phylogenetics Research. Of course, by trees we are talking about phylogenetic trees, and I just realized we could have titled it All Tree No Shade, but we'll continue with our title here, working title, These Trees Cast No Shade. So I'm Brian Minalga from the Office of HIV/AIDS Network Coordination and the Legacy Project. I'm happy to welcome you all to our webinar today, where we'll be talking about this technology that's being used in the context of HIV research. Some of you may be asking, phylogenetics, what is that? And we'll define that, we'll walk you through it, so everyone will be able to explain what that word is by the end of the webinar. For those of you who attended part 1 of the webinar last month, we'll also be taking a bit of a step back from the topic of that webinar, which was more focused on the public health arena. And in today's webinar, we'll be talking about how HIV sequencing is being used not in the public health arena, but rather in the context of HIV research. And there are some really important differences between those two arenas, public health and research. One of the most important of which, of course, is the concept of consent and informed consent, which was a key word from our previous webinar. So pay close attention to that in today's topic. We will be hearing from 2 experts today, one bringing expertise in HIV clinical research, and another bringing expertise in ethics and community engagement as a person living with HIV. And before we start with the webinar, I just also want to acknowledge that today is National Transgender HIV Testing Day. So we wanted to give a shout out to all the transgender and gender nonconforming people across the country who are participating in this day, whether you're getting tested yourself, helping a friend to get tested, or working at a testing related event. And of course, thank you to the cisgender allies helping out with testing today. We couldn't do it without you. So just a few logistics about the webinar. This is being recorded and will be available on our Facebook page and then our website, but the easiest way is probably through our Facebook page, which is right there at Hank Legacy Project. And when I'm done speaking here in just a moment, I'll post the link to our page in the chat box so you can just follow us right away. There will also be a brief survey following the webinar, so please fill that out at the end. That would help us a lot. And lastly, this is intended to be an interactive webinar, and so you can use the chat box in the lower left-hand corner of your screen. You'll have a chance to ask these 2 experts questions and make some comments. So go ahead and feel free to use that chat box throughout the presentation. Just a reminder that this is a 4-part series. Today is part 2 of that series. We did part 1 last month on March 27th. The recording of that is available on our Facebook page. And there are 2 more parts coming up, one on April 25th, where we go back to talking about public health and molecular HIV surveillance. And then we'll finish in May, on May 15th, with a community town hall webinar. So I'll also post a link for registration for those subsequent webinars as well on the 25th and May 15th. This is a partnership webinar series between these organizations, the United States People Living with HIV Caucus, the Positive Women's Network USA. I'm Brian Minalga from the Legacy Project. We have the Zero Project, Thrive SS, the Reunion Project, the International Community of Women Living with HIV North America, and Positively Trans. So thank you so much to all of our collaborators. And with that, it is my great honor to introduce our 2 presenters today, Carl Dieffenbach, the Director of the Division of AIDS at the National Institute of Allergy and Infectious Diseases, and Venita Ray, the Deputy Director of Positive Women's Network USA. So with that, I will hand it over to Carl.
Thank you, Brian. It's really a pleasure to join all of you today to be able to discuss the subject of phylogenetics and try to make it approachable. And you'll see why this kind of research gets me interested and excited as we go through this. Next slide, please. So this is what I'm going to try to cover for all of us today is what is phylogenetics? How are phylogenetic analyses being applied to HIV research? How might phylogenetics analysis benefit communities most affected by HIV? And then at the end, just touch on some ethical issues that HIV phylogenetic research raises, and how are researchers addressing them. That'll be, I imagine, a topic for discussion when we get to the very end. So basically, I have a list of different subjects that I think are worthy of consideration. Next slide, please. So if we think about phylogenetics, we have to think about it in terms of what where it arose and why this is important. And fundamentally, what phylogenetics is defining evolution through DNA sequences. So if we think about a molecular clock, or a clock that is ticking with every moment of existence of life on this planet, the DNA that makes up the genetic material of those organisms has been evolving since the beginning of time, and the evolution of DNA is relatively constant across time and lineages. And what a molecular clock allows us to resolve is the history of species, the timing of events, the relationship of species. And this really got its start from the analysis of proteins, the actual molecules that do the business in cells, as opposed to the genes that encode them. If you look on the left-hand side of the screen, you see an old-fashioned paleological tree of vertebrates. And just to give you a perspective about what we're talking about when we think about the evolution of human beings, if we compare human beings in terms of the sequence homology, the amount of identical DNA present in a human compared to a mouse, for example, it's 97.5% identical. So 2.5% of our DNA separates humans from mice. If we compare our sequence to zebrafish, it's 85% difference. And if we compare our sequences to fruit flies, there's 36% homology. So even if the fly, without bones, without being a vertebrate, has some significant sequence homology with humans, and that tells us about the function of genes. On the right-hand most part of the screen is a really important point I want to make early in this presentation, and that is as we compare the molecular clock for, say, humans and chimps and mice and zebrafish, it's on a scale of millions of years. However, when we think about the evolutionary scale of HIV, the time scale is in weeks to years, not millions of years. Next slide, please. So let's talk about applying phylogenetics to HIV and its evolutionary history that we can define by studying the change in HIV DNA. And I want to take a big step back and talk about HIV disease from the perspective of phylogenetics. And that is that one of the greatest strengths that HIV as a disease-causing agent has is its ability to change. It's like a shapeshifter. It changes its sequence, it evolves very quickly, and in so doing, it evolves in such a way that it evades the immunity that humans can traditionally generate against other viruses or pathogens. But it's important to consider this here is by using phylogenetics to study HIV evolution, we are turning the virus's greatest strengths into a tool to help us as scientists and community to first understand HIV and then to defeat HIV. Next slide, please. So just how fast is fast? So the words on the left-hand side of the screen there describe just how fast it is from the perspective of the HIV genome. And the HIV genome is made up of 9,200 base pairs. That's its nucleic acid sequence is how we refer to it. The critical enzyme that helps make copies of the HIV is called reverse transcriptase. And it is what we refer to as an error-prone enzyme. And it makes a mistake every 1,700 bases, resulting in approximately 5 changes in the sequence with each time a copy of a virion is being made. In the course of an untreated HIV infection in the human body, there are, on average, 45 million copies of HIV made per day, and this is an untreated infection. And therefore, what we can, if you start doing some calculations on that, that leads to a change in every position in the nucleic acid in the genetic material every day in an individual. So that is what we would call to as very rapid change. In addition, this results in a lot of mistakes, and that's one of the reasons there's so much defective nucleic acid present in people living with HIV. And this explosive growth plus the error rate leads to evolution. Now, if we want to see what that looks like visually, in the upper, if we look at the Medscape slide that I downloaded there, the first little set of squiggles is the size of the variation in influenza. Now, you all are aware that every year influenza changes to a point where they have to make a new flu vaccine every year. But in the global pandemic of 1996, that little tiny squiggle was the size of all the genetic variation that was generated globally for influenza that year. The next one down, and the squiggle is actually is bigger for the next one than the flu, is HIV evolution in a single individual 6 years after infection. The next biggest squiggle is the Amsterdam cohort as of 1991, representing everybody who had been enrolled in the Amsterdam cohort at that point and had their nucleic acid sequenced. Compare that to what was present in the Congo in 1997. So you can see graphically just how fast HIV evolves and changes. Next slide, please. So importantly, because HIV's rate of change is so consistent, we have been able to use this and have developed methods to look at the two examples we're going to talk about today. One is a public health example, which is looking at what happened in Scott County, Indiana, and the other is a study, HPTN 052, which looked at linked and unlinked transmission events. We're going to talk about why that matters and also take head on the question of directionality. Next slide, please. So on this slide are 4 panels, and they describe graphically what has been accomplished in the evaluation of the outbreak in Scott County, Indiana, which was reported on several years ago. Importantly, we have to keep in mind that Scott County had become a desert, become a desert where there was no healthcare providers. So it was a healthcare desert. And you can see starting in November of 2014, where the initial diagnosis was made, and over a period of the next, from November of 2014 to November of 2015, over a short year, they'd identified approximately 180 HIV positive individuals and been able to then help to bring it under control through syringe exchange, testing, and counseling. And the way we know that these were all related is in the middle panel, where you can see what's blown up in red from that one red arrow, is that everybody, that represents a sequence analysis of everybody who became HIV positive in Scott County, and you can see just how related the viruses were. On the right-hand most side is what we call a hairball graph, which shows the relationships of the people in Scott County who had partners who were either HIV positive and negative. And it's important to look, as you look at this, you can see under connection type, the people were either connected by whether they shared syringes or shared sexual contact. And you can see, even in the case of individuals who had syringe sharing, not everybody who shared syringes, even with a person with HIV, became HIV infected. So this is an important graph, but I want you to keep in mind that this was all very retrospective. And one of the challenges we face that you'll hear about at the upcoming webinar is how can we take this data, put it in real time, so when there is an outbreak, we can get in, as CDC will be discussing, how we can get into an outbreak in an appropriate way, get people tested, get people on therapy, get PrEP, and needle and syringe exchange into a community so that we don't have this kind of an outbreak persisting. Also, keeping in mind that if there had not been specific, so those of you who know Greg Gonzalez at Yale have probably seen his article where he described in detail why Scott County occurred. And it was because of actions by the then governor of Indiana closing the only care facility in Scott County because it also offered contraception services to women. So Greg is pretty clear about who's to blame for Scott County, Indiana. And if you haven't seen his article, I would encourage you to look at it. Next slide, please. Switching gears, let's talk about HPTN 052 and the question that HPTN 052 wanted to test. The question was, can provision of treatment and getting people virologically suppressed in a serodifferent couple, could that have an impact on HIV transmission? So in the couple study, we enrolled 1,763 serodifferent couples. They were randomized to start early or start immediately. And in the course of the study, there were 78 transmissions, 55 in the delayed arm and 17 in the early arm. But the important data I want you to look at here is the number of linked infections. In the delayed arm, there were 43 linked infections, and in the early antiretroviral therapy arm, there were 3 linked infections. Subsequently, the study team has gone back and analyzed those 3 linked infections to be able to show that they came from viremic partners. Next slide, please. So because the focus is on link transmission, because that's where we get the precision in our analysis, HPTN would not have been able to HPTN 052 would not have been able to demonstrate as much interesting data if we were comparing 55 transmissions versus 17 transmission events, but instead we were comparing 43 transmission events to 3, and on closer inspection, 43 to 0. And I want to remind everybody that this was one of the sentinel events in establishment of the U equals U principles. Subsequently, other studies have gone on to amplify the HPTN 052 data to a point where now U equals U is a very well recognized platform that makes it so that HIV treatment provides not just benefit to the individual, but also offers a way out of self-stigma in a way that is beneficial to the person who's receiving treatment. Next slide, please. So now I want to talk about, can phylogenetics infer directionality? If you're thinking about it, the study from the standpoint of study of evolution, directionality is obvious. A zebrafish is lower on the phylogenetics totem pole than chimpanzees or human beings. But there's a quote in there, and I want to read the quote to you so we're all on the same page with this. That from an editorial that when a specific article appeared in the Journal of Infectious Diseases. For many research questions, directionality is not central because identified potential transmission pairs or clusters is sufficient for their assessment. Nevertheless, for detailed transmission studies of early pathogenesis events, particularly for legal cases, correct prediction of directionality is of high importance. I think that is the major concern that all of us have as we study phylogenetics, that somehow directionality will be an important part of where we're headed. And because of U equals U, we can really talk about decriminalization and eliminating this concern. But now, are we essentially going backwards in time? And I think it's important to take this head on and be able to continue to talk about this concern. So at this point in time, the next slide is going to be a quiz that we would like you to answer as part of your participation in this webinar today. Can you please read the question, which says, can phylogenetics infer directionality of HIV transmission? And this is the pre-lecture quiz where you should vote your opinion on this and submit your answers. And now, just like Alex Trebek, I'm going to give you a minute or so to answer the question. Please go ahead and answer the question.
And I'm just monitoring how many folks we have. Looks like we've got about almost everybody, so we'll give it just another couple seconds here.
Yeah, then you're going to switch the slide there, so all will be revealed.
All will be revealed. And what I have coming in is very interesting already.
Well, that's interesting. I'm fascinated to hear to see the outcome of this.
It's very exciting. All right, so I'll just give it maybe 10 more seconds, and then we'll close.
Sounds good, Brian.
All right, so let's close the poll here, and I'll show the results. Interesting.
So 48% of people feel that directionality can be inferred of HIV transmission. 13 don't know, and 37% are a no. So keep this in mind, and we'll come back to this slide in just a moment. Next slide, please. So let's go to this. I think you skipped one. Can you go back a slide, Brian? Oh, sorry. Go forward. My mistake. So if there's any HIV transmission study where we know directionality, it's from HPTN 052, because remember, it enrolled 0 different couples. So what the study team did is they took this as a model data set and then modeled it to see if, based on the sequence, they could determine accurately, looking at this data in a blinded way, the directionality of transmission. And importantly, what they found is published in this article by Rose et al. in JID in 2019, is directionality was predicted correctly for only 61% of the index pairs for both trees and sample sets. So current methodologies are not able to reliably predict directionality. Let's go back to our poll. So the people who said no were correct. With the current technologies, we are not able to infer directionality on HIV transmission. So I think that's an important misperception that we want to be able to correct, because like for most research, we're not interested in directionality. And I think that that's where, when we get to the public health section at the next webinar, I think that'll be important also to be listening for the CDC to discuss this. But the protections are different in these situations. Next slide, please. And next slide, please. So I now have a couple of lists of additional items to consider as we think about phylogenetics in research. So next slide, please. Let's talk about the importance of human subjects protection. So there are specific actions that could be taken in terms of, if you have a DNA sequence data set like the 052, it could be completely de-identified and anonymized, so all you know are the sequences and the ones that are available. The researchers should also consider specific wording in the informed consent, and whenever possible to use previously collected data. Also to be aware that state law needs to be considered when using public health surveillance data for research purposes. That's not the subject of today's lecture, but I think that's important as we go forward, that we're not just governed by federal rules, but also state rules around the country. Next slide, please. For community considerations, so these comments came out of a publication that came out of a meeting here at the NIH where there was a community consultation with researchers to generate these lists. So that's where I didn't sit around and make these up. These are actually from a publication that was put together in consultation between an ethics group and community. Targeted community engagement needs to be considered when specific communities with specific identifiable population groups could be impacted. I think that's very important. It's always, community engagement needs to be first and foremost in all types of research, but for this it's particularly important. Start community engagement. This is almost like community engagement 101. Start community engagement early in the research development. That's obvious, so community can have input. Researchers should be aware that other populations may emerge with the onset of the projects. There may be subgroups that are identified that people need to be cognizant of. Next slide, please. And when it comes to communication of results, we need to be very sensitive to the wider effects and implications when reporting findings. Research findings need to be disseminated in accessible formats, and appropriate educational context must be included when communicating this type of phylogenetics research. Next slide, please.
I think that's your last one, Carl.
That's one. Okay, thank you all, and I'll turn it back to Brian.
Great. So before we go to the next presenter, there are a couple questions, and I think for the purposes of time, we'll just take the two that are here. If other folks have questions, feel free to type them in, and we'll get to them at the end of Venita's presentation. But there are just two questions for Carl right now, and one of them was just asking to see that Scott County slide again. So I'll put that back up here.
So these are direct from the New England Journal of Medicine article that was published a while ago. And Brian, you're going to make the slides available on the Hank portal?
Yes, slides will be available on the Hank portal and public website, and also our Facebook.
So whoever is interested in this slide will be able to download it from my presentation.
That's right. And then the other question is coming from Bill. And Bill wants to know about directionality. So I'll put up the slide that he's referring to where it says reliably. And Bill is asking if we can define reliably.
So reliably in this context, it would be that you had to be between 95 and 99% sure that you had the right answer. So if you were, that would be a reliable level of prediction of directionality. So in this case, where you're wrong 3 or 4 times out of 10 is not sufficient to say these are related pairs. So that's the difference. It's the difference between an error of 1 in 100 or 1 in 50 versus 4 in 10. So 4 in 10 is significant.
Great. So especially if you think about the context of a criminal case, that wouldn't be, we wouldn't have the evidence and the scientific standing to be able to use this type of direction.
Definitely not.
Yeah, great. Thank you. I see a couple more questions coming in, so thanks for those. Keep them coming. We will get to those at the end of Venita's talk, but for now we want to make sure to give Venita her time. So this is Venita Ray, Deputy Director of PWN USA. So go ahead, Venita.
Hey, good morning or afternoon, depending on where you are, everybody. And thank you, Brian and Carl. I appreciate your presentation as well. I felt strange being introduced as an expert. I'm a person living with HIV, but for the last couple of years, I'll share with you how I got involved in phylogenetics and actually learned to spell it sometimes. So thank you for having me as well. Next slide. And if you don't know, let me just say this, Positive Women's Network, we are a constituency-based social justice organization of women and trans folk living with HIV. So we work on policy issues and leadership development and electoral organizing for our members. I guess the reason why I'm here, I got involved in the phylogenetics work a couple years ago when invited to participate with an NIH work group where I will give NIH some credit, and I'm glad Carl and Brian kind of delineated this distinction between research and public health, and up until that point, I did not see them differently, nor did I understand how things were treated differently in those different arenas. But they put together a work group and decided to develop some ethical guidelines for researchers and use of phylogenetics in HIV-related research to examine those potential risks, social harms, and related ethical harms. One of the things, and I think came out with Carl's presentation, and I heard a lot around the table, even though this was approximately maybe 40 people around the table, 35, there was only a couple of us that were living with HIV that I'm aware of that were around that table. And because this science applies, it looks like the evolution of species, and it's not just referring to humans. There were a lot of references to animals and different things, and we were really there talking about people living with HIV and how to, how to develop guidelines related to this research when it involved people living with HIV. So even though it's a broader science and the study of species and evolution, we really have to remember we're talking about people here, and that was one of my big concerns, and appreciate NIH pulling us together to talk about how to apply this science to people, even though it's used in a broader context. So next slide. So it started off with an in-person meeting in 2018 and divided us into work groups. People self-selected, and I chose to work in the community engagement arena because of as being a person living with HIV, really wanted to have some input in how the community was engaged around these issues, or should be engaged and educated. And one of the things that Carl highlighted before, there are misperceptions about the science and how it's used, even to the point of when we talk about reliability, the question asked, can you infer directionality? And I see that as a little different than reliability, being reliable in predicting directionality, a little bit different. And so I wanted to be a part of that, and I also joined the legal issue groups because I think I was invited to the work group because I've been involved in a lot of the HIV criminalization work, and the issues of directionality and what could be proven or not proven or what folks know was really paramount in my community and caused a lot of fear. So those are the two frames I chose to focus on this work. Next slide. So there is a paper coming out, and again, like I said, I gave NIH a lot of credit for the way that they were thoughtful and bringing these folks together and not moving it so fast, and really listening to the concerns and really trying to address them. And it presented a really good opportunity for education for me. So, and some of the things I want to highlight around community engagement, and Carl mentioned also the degree to which, community engagement is necessary for research. We know public health, we're always talking about there should be some engagement that doesn't always happen or happens in a kind of different way that we would call engagement. So we talked a lot about that. It is necessary for all research projects that impact people living with HIV. The love of science in and of itself has sometimes unintended consequences on those people on the ground, since we are talking humans. So community engagement is necessary. The issues that kept coming up was the extent, to what extent community engagement should be had. Are we going to the planning councils, prevention planning groups? things like this, and I really stress, and others did, that community engagement is broader than those bodies, you know, when you really want to educate people about what's going on. And we did talk about the level of community engagement depended on the design of the project, the type of project, but community engagement in and of itself is not one-dimensional, one-size-fits-all. It really should be broader than the usual kinds of folks we go to and educate community, which is why I think that the poll that was done was so off, because we believe, you know, there's a perception out there because there's not a lot of education about what the science really can show. Additionally, the potential impacts to one of my points in this is that those potential impacts that researchers or others use to decide when community engagement should be had or how much should be viewed from the perspective of the community and not just the researcher who says, I may not think this is going to have impact, because there's impact to communities that folks don't always understand or know unless you are a member of that community, and some of them are unintended, of course. So I think that when deciding the type of community engagement and the extent of it, it should be viewed from the perspective of the community when you're looking at what those risks, harms, and potential impacts. And similar to Carl, I think the engagement with community should be started at the start of the research, not design it. Or one of our issues we have with molecular surveillance is kind of out of the barn, and now we're kind of being brought in and talked to. So, and then this idea of lack of knowledge about the use of the science, which I think the polls show, when we don't, you know, educate or really bring community along at the same time that we're doing the work and distinguishing the difference between the work in public health and research, we have a lot of fear, justified fear as well, and some fear that's not justified because it's just we don't have an understanding and knowledge, which I did not have. Like I said, when I first started in this work. Next slide. From the phylogenetic standpoint, I kept saying that word over and over because I couldn't believe I could even say it, let alone spell it. So I got involved in the legal issue side. I happen to be an attorney who I don't practice anymore, but heavily involved in criminalization. And some of the key points I'll talk about here is that, and Carl mentioned too, there's all kinds of different sets of regulatory standards, state laws that impact who can get access to information, data security, when is informed consent required or not. The distinction between how research informed consent is handled, which is typically required, and what happens on the public health side. I hadn't even heard of the certificates of confidentiality that are required for research. And then there were issues that came up of what they call secondary use of public health information. So this is public health information that was derived under public health standards or state laws, but then researchers may use it for research purposes, and so there just became kind of a multiple kind of legal issues that could come out of the use of the data and the science. And then the second issue for me around the legal issues was the degree to which this information could be used in a criminal or civil case against an individual who's alleged to be the source of a HIV transmission event. Now, whether you can reliably predict, infer, if the belief is on the ground that you can and that folks can point to you, then the belief is real to them, perception is real that this is the truth, because folks do not know. And then when you look at a criminal versus a civil case, the burden of proof is different. So, and then when we look at in legal issues that could refer to sex workers, it could refer to not only transmission, but all kind of folks that are coming from communities that are heavily impacted by HIV that come into contact with the criminal justice system for all kind of reasons, that this just adds another layer of fear, another layer of complication, another layer of worry and concern for the HIV community. Next slide, please. So some of the other key issues for me and what came up was this issue of informed consent. I didn't know until I started this work that it's required for research purposes, even though, like I said recently, there are still issues that arise because research could go in a direction it didn't think, or we could come up with a potential harm or a risk or a consequence that wasn't anticipated. Informed consent is not required in the same way for public health purposes for use of our genetic material. So this issue, if some of you were around when we did our first series, it was called Without Our Consent, is because we believe we never consented to the use of our genetic material in this way and didn't know that it was happening until a few years ago. So the issues around informed consent in a research versus a public health context are different, but we need to address it when we're looking at this science. Again, scientific advances, which is, they've been working with phylogenetics for years, could result in negative impact on positive communities. That, science, when you're doing something for the love of the science, it's not always we're not always thinking about what will happen on the ground and what that means, because we always hope science advances us, right? And that gets me to my point of when does the benefit outweigh the risk of harm? Clearly, 1996 and protease inhibitors changed the course of life for those of us living with HIV. So science is definitely necessary, but how do we decide and when does the benefit outweigh the risk of harm? Or when does the risk of harm outweigh the benefit of what's being studied, researched, and having this technology used best? So those are key questions. And then cluster data, which is the way that this science is used to show that there are linked transmissions or transmissions that have similar characteristics at the public health level is no longer anonymized. There could be names associated with that, and in the beginning we were told there's no names associated with it. Well, in order to get the public health benefits that have been talked about, you need a named partner or something to do the linkage work that folks are talking about we need and things like that. So it is not anonymized as we thought at first, and again, that's a clear distinction between the public health and the research side. Next slide, please. So lastly, I think this is my last slide. Overall in a lot of our communities and happen to be from Black communities, myself, and living with HIV and other communities, there's a historical distrust of science and research. The advances are great, but, the cost to some of our communities for those advances, when you think of Tuskegee or you think of other types of times that science, was questionable in the ways that it came about, in its advances. There's potential negative impact on communities, again, that need to be weighed against scientific advance. Again, benefit versus harm. There's a belief that science could evolve to identify the source of the transmission. I know Carl and I've read other reports that say we can infer or reliably predict directionality, but there are those of us that believe that the science will continue to advance and that will one day happen. And even if it hasn't happened yet, there's a belief that it's already happened. So the science will continue to evolve, and we believe that will occur. There's a general lack of knowledge that our genetic material taken from our bodies was being used in this way. I think I've already mentioned that, again, without our consent. And from a public health context, that there's a belief that you don't need our consent to use our genetic material in this way. I think there's still some privacy concerns of people living with HIV that should be considered. From a public health perspective, the good of the public sometimes outweighs the good of the individual. And for those living with HIV that's heavily stigmatized, there could be drug users, sex workers, or other just heavily policed, marginalized communities. Those concerns are multiplied, right? And so when lack of consent, lack of informed consent, privacy rights, all should be considered when developing guidelines and considering the use of this science. And, lastly, I would say we want real input in the way that genetic material taken from our bodies is used. Real input, say so, informed ability to know that it was done. And up until now and in this last couple of years, and I appreciate people like Carl and Brian and others that are bringing this information to the community to let us know how our information is being used, but which should have happened in the beginning so that we were all on board and understood the consequences from the beginning. I think that might be it. And I always want to remind folks that we're talking about humans here, and the science of phylogenetics is about species. But as it relates to HIV, when we're talking clusters, let's replace that with people living with HIV so that we know we're impacting people here. And next slide, I think that's it for me.
Yeah, that's your last slide, Venita. Thank you so much for your presentation. And I must admit, I feel extra nerdy today because I am living for this conversation just after hearing both of your presentations. So I want to be sure to thank both Carl and Venita for amazing talks. We do have a number of questions already, and we have about they're all good questions. We have about 13 minutes left to have some discussion with the audience, so folks can continue typing in questions. The 3 questions I have right now are all actually about this question of directionality and this concern with criminalization. And so before I start to kind of ask those questions, I think one thing that might help us have that discussion is to help, again, as Carl and Venita have done, to delineate between these 2 realms, these 2 arenas of public health, use of sequencing HIV genetically, and then also what's happening in research, so in clinical research in studies. For example, when people living with HIV are participating in a study of a new HIV treatment or a potential cure for HIV, or even when HIV-negative folks are participating in HIV prevention trials with the possibility of seroconversion during that trial. So that's kind of the realm that we might be able to discuss this issue of criminalization, because next week, just to remind folks, a week from today on the 25th, we have part 3 of this series with Dr. McRae from the CDC and also Nana Khanna from PWN USA to talk more about the public health arena and some of the public health and human rights implications of molecular HIV surveillance. But if we can maybe turn our attention a bit to something that I think doesn't get as much attention as the research and how this technology is being used in research, maybe we can ask the questions there. And maybe my question to both Venita and Carl, in your experience doing this work, what access do folks have? What access, for example, would a prosecutor have to research data whereby HIV has been genetically sequenced? Is that a possibility?
This is Carl. That's a really scary question, and I understand that it needs to be taken head on. But, from the perspective of going back to something Venita said about control of the data and something I said about, when possible, and now anonymizing the data, you have to if you've worked it out in such a way that so basically they would, let's just take this to a logical place, that you go and you serve the University of Washington data center a subpoena saying they want all their sequences because they want to look at this. I think the answer would be that is they're not able to get that data because it's not it would not be within their, what would be the crime that they would be investigating. And I think that it would behoove us to keep in mind that as we go forward these eventualities and protect through the steps that I outlined about anonymization, maintaining informed consent, and be absolutely clear about the use of the data. So the two examples you raised, Brian, we're going to, as we complete the AMP trials, we do intend to sequence the virus from people who become HIV positive for the purpose of looking at whether the antibody had an impact on the sensitivity of that virus to VRC01. That's a process called sieving. And I think that that's very clearly laid out in the informed consent. And your second example of use, what was it? You had a different type of research. Oh, cure. The issue on cure research. So if we're going to have people that receive treatments for they're virally suppressed, and they are then treated in such a way and then undergo a structured treatment interruption, it's very important to understand the structure of the virus, its nucleic acid structure that emerges from an individual who is undergoing a structured treatment interruption, so that we know how many viruses and what's happening. But those are really good examples where informed consent and would be absolutely critical. But in that situation, particularly the structured treatment interruption, the participant is an active partner in this because they have to come back and get tests, and they will know when they, if and when they rebound, and they will want to stay linked with their doctors so that they can go back on therapy when they meet the restart criteria. So that's where I think we have to continue to talk about the partnership between researcher and study participant, and make sure that the informed consent spells out exactly what is going to happen and how that information is going to be used.
Great. So informed consent, really important. Venita, I know you had a moment on the slide here about certificates of confidentiality. That might be something to bring up, but just wanted to see if you had any thoughts on this as well.
Okay. Your question was if a prosecutor wanted to subpoena any of this data that had been used or gathered for research purposes. Was your question?
Yes.
Okay. Yeah, and so that's why this has been so interesting to me, because I'm glad Carl answered first, because in that context, there are more protections, and the certificate of confidentiality that exists in a research context that may prevent that access to that information doesn't exist, to my knowledge, in the public health context, other than state laws or other protections, HIPAA and things like that, although HIPAA allows subpoena for law enforcement. So certificate of confidentiality is something I learned about for research purposes that could be used, but that it could be used to deny access to that prosecutor. But I wanted to also ask the question, or just make the mention of, if that same prosecutor, if the original data was developed through public health and it was being used, secondary use by researchers, that same prosecutor may have the ability to go back to public health and try to get that same information.
Okay, great. So thanks for that. There are a few more questions kind of related to this issue. Nicole is asking Carl, is the research being conducted with the purpose of trying to determine directionality of transmission, and if so, why?
No, it is not.
Okay, nice easy yes or no question. There's a comment from Karine who says, we will need community and patient-friendly materials and informed consent forms for phylogenetic testing. So just a comment from Karine.
Wow.
But again, let's come back and make sure that we're not undertaking phylogenetic testing for the sake of phylogenetic testing. It's within a research context to answer a question. And when you talk about the public health next week, it's for different purposes. And I think that this is something Venita said is the public health good. Everybody who is starting antiretroviral therapy is supposed to get a nucleic acid sequence done. Why? This is important. The reason they get it done is they need to know if they are carrying mutations in their virus that makes it so the drugs don't work. And so that data is stored and might become available for phylogenetic testing, but it's a piece that is of benefit to the individual that a doctor generates. And I think we want to make sure that we continue to talk about some of the strengths and advantages of getting sequencing done, such as the last thing you want to do is start with a drug if you already have resistance to it. So I think there's some tremendous advantage to this. I think there's a lot of education, and that's why this is a very important webinar today, that we need to be able to talk about additional uses of how sequencing is improving care for people with HIV. And it's not, we're focusing on this whole thing with directionality. And I think that the point about that we can't, so I actually don't, so somebody said, this is something Venita said, maybe someday the research will improve to a point where you can predict directionality. And I guess, the part of me says, I understand what you're saying, but a sequence is a sequence. You're not going to improve on the core piece of data. And there may be more of it, and the computer program may get better. But unless we understand some significant rules, I mean, I don't see this as being an area where people will have an active interest. And I don't think that it's going to actually get to a point where it can be 99%, even with the best tools available. There's always going to be doubt, simply because there's just so much noise in HIV. But I'm an optimist, and you're probably right. So please push back.
And I want to just make one comment to that. And so we do need the initial testing to determine what medicine we use. We make that deal with the doctor to get it, and we don't know that there's going to be further use of that genetic material for something else that could benefit the public good. That were the decision that was made between us and our doctor to do it for to determine the best medicine, and that there was not knowledge that it would go further than that. So that's one thing. The second thing is I remember speaking about a year ago to an attorney who does this work and is aware of it, and became aware of a defense attorney in Florida who was defending someone who's being charged with transmission and wanting to get the phylogenetics research to try to get their client off, and we were like, don't, no. They didn't really understand how to use it, but it could end up harming the person, and I'm not sure what happened in that case, but we don't understand its use. And for people who are living with HIV, and we see criminalization cases every day, that's why directionality, whether you say phylogenetics, that's not the point of it. that's why it's such a big fear, the threat of it, the thought of it, and that's why the poll, I think, also went the way it did. But we do hope to educate more, and I still believe that science is going to evolve.
I agree with you. I think let's just come back to the sequence that is stored when a patient gets a test in a doctor's office. What gets ultimately stored is completely stripped of all personal identifiers, and all it is a sequence from, and that's all that's available. It's not, there's no personal identifiers left. And that is one of the key points on one of the slides I made, is to strip out, use anonymized data, and use anonymized data. That's, that's what we should be using to the greatest extent possible.
Great. Well, we are out of time. Thank you so much to both of the presenters today, Carl and Venita. We really appreciate you giving your expertise. There are still some questions coming in kind of about this directionality issue. We're going to go ahead and punt those questions to the next webinar with Eugene McRae from CDC and Nana Khanna. So, this is, I think, more the questions are a bit more related to the public health realm than to the research realm. As far as the research, Venita mentioned, this working group at the NIH, which had taken on and, did take on this issue of legal issues within research, and a publication will be coming out. We're currently seeking publication for that paper. Venita and I are both on that working group. And so I'll make sure that when that paper is published, I can go ahead and send it or a link, or make sure that the folks who joined this webinar today are aware of where that paper is located, so you can see more about how this issue plays out in the research realm. But once again, please join us next week, Thursday, a week from today at 11 Pacific. I put in the registration link right into the chat box, and I see some folks have already clicked on that and gone ahead and registered. So please join us next week. And thank you to all who participated by asking questions, and your attentiveness to our presenters. And of course, once more, thank you so much to Carl and Venita for your expertise today.
And thank you, Brian.
Yes, thank you, Brian.
Appreciate you. All right.
All right. Take care, everybody. Bye-bye.
Bye-bye.